In this video lecture, we're going to examine some basic hematology procedures, and our goal is to explain those procedures, specifically the complete blood count, hemoglobin measurements, hematocrit or packed cell volume, and red blood cell indices. A complete blood cell count includes a lot of different tests. It's a very routine procedure performed in the lab on a lot of different specimens, and it gives you a lot of information, including your hemoglobin, hematocrit, red blood cell count, red blood cell morphology, shape and size, red blood cell indices, white blood cell count, white blood cell differential, and platelet estimate. In our discussion today, we are going to focus on hemoglobin, hematocrit, red blood cell morphology, and red blood cell indices. So let's take a look at some of these tests. When performing a complete blood count, you want to use anticoagulated blood, and the anticoagulant of choice is potassium EDTA. This is in a purple top evacuated tube, and what it does to prevent blood clotting is it chelates the ionized calcium which is floating around in the blood. Calcium is one of the important factors that you need in order to stimulate a blood clot. And so if you remove the calcium, a blood clot can't be formed and the blood remains anticoagulated. This picture here kind of shows what happens in chelation. Basically, chelation is when you bind up a molecule and make it inert. So imagine that this would be the little calcium molecule right here being chelated by the tripotassium ethylene diamine tetraacetate. That's what that EDTA stands for. So pretty lengthy. As long as you know potassium EDTA, I will be happy. This is what it does. Now, in a tube, 2 ml tube, you will find 3 milligrams of this potassium EDTA because you need 1.5 milligram for every 1 milliliter of whole blood that you have. And the ratio is really important. You want to make sure you have just the right amount because if you have too much of this EDTA in your tube, you can actually cause the red blood cells to shrink, which, as you saw before, part of what we're looking at in a complete blood cell count is the size of the red cells. And if they are shrinking, not due to the patient, but due to the anticoagulant, we're going to get some results that aren't really applicable. They're going to be false. So it's really crucial when you're drawing blood for you to make sure you fill the tube completely. Otherwise, it could affect your test results. Now, we're going to start looking at hemoglobin measurements. And the way we measure hemoglobin, we touched upon in the other video, and that's the hemoglobin cyanide method. What will happen is a sample of blood will be drawn, and added to that sample is a reagent known as the Drabkin's reagent. And it has a few different things within it. One of the things it does is it has a red blood cell lysing agent in it. So we actually rupture all of the red cells so that we can expose the hemoglobin. And then within that Drabkin solution or Drabkin's reagent is potassium cyanide and a few other chemicals. And what will happen is the potassium cyanide will bind with the freed hemoglobin. After you mix it with the specimen, the binding will occur and you will actually form a pigment known as hemoglobin cyanide or cyan met hemoglobin, and that pigment will be able to be measured by colorimetric procedures using a spectrophotometer. So what the spectrophotometer will do is measure how deep the color is, if it's dark or if it's faint, and it will give you a readout as to the absorbance of that sample, and then you can correlate that absorbance to a concentration of hemoglobin. So every hemoglobin molecule that has been freed by the lysis of the red cells will be bound by the potassium cyanide and will form the cyan met hemoglobin, and that will cause a color change, and that color change, the intensity of that color change will be measured by a spectrophotometer. You can see here, this is just, you know how I love physics, but this is just a little bit of physics, basically showing you have a light source that will shine through your sample and your absorbance will read out. And obviously the darker the sample, the greater amount of light it's going to absorb, so the greater the absorbance will be. And the higher that is, that means you have more hemoglobin in your blood sample. Normal reference values for hemoglobin measurements are in an adult male, 13.5 to 17.5 grams per deciliter of blood, and in an adult female, 12.0 to 16.0 grams per deciliter of blood. Now obviously women, when they menstruate, will lose some blood and therefore their hemoglobin will be lower, Generally, females have a lower body size, which accounts for a lower blood volume, which also relates to the hemoglobin level. And testosterone is one of the things that stimulates red blood cell production. So generally, males have more testosterone in their system, which causes an increase in red blood cell production and thus hemoglobin within the red cells. So make sure you're familiar with these reference values. And it's different for children and infants as well. I just like you to know the adults. Next, let's look at a hematocrit, or also known as a packed cell volume. And basically what we're measuring in the hematocrit is the ratio of packed red blood cells to whole blood. Now, a packed red cell is implying that the sample we've collected has been centrifuged. And you can see up here, this is a microcentrifuge. And what will happen is we'll collect some blood. You can collect capillary blood, or you can draw blood into a tube and then fill a capillary tube with blood. And you put it into a microcentrifuge and you spin it, and what will happen is the red cells will be forced to the bottom of the tube. And then you'll notice also there's that buffy coat that we're familiar with and the plasma at the top. And we look at this and we see, okay, what percentage of this whole blood here is taken up by packed red cells? There is a normal percentage that we should fall in, and if it's abnormal, we report that out, or we report it if it's normal as well. But we're really looking at how much of our blood is actually cellular. We can, from the hemoglobin measurements that we got prior, you can estimate what the hematocrit is and make sure that things are making sense. So if you take your hemoglobin measurement that you got using the Drabkin's reagent and spectrophotometry and multiply it by 3, that should report your hematocrit with plus or minus 3 units correctly. And since this is a ratio, there's not actually units on a hematocrit. It's just a percentage, essentially. Normal reference values for hematocrit, as you can see, are higher in males than females, which correlates the same to the hemoglobin measurement we just talked about. But in adult males, it's 41.5 to 50%, and in adult females, it's 36 to 45%. Now, the red blood cell indices are a little bit different than the tests we've been talking about. The hemoglobin and hematocrit will measure entire volumes of blood, how much hemoglobin is in this 1 milliliter of blood. Whereas the red blood cell indices are going to look more specifically at an individual cell. And so this is really helpful when you're diagnosing anemias or other issues where the cells are misshapen in size. And the 4 that you see here, the MCV, MCH, MCHC, and RDW, are basically measured using hemoglobin, hematocrit, and red blood cell count numbers. Oftentimes now they're done electronically, and so that makes it a lot easier. There's a lot of error when it was done by hand, but we can see that the fancy machinery we have in the lab now is capable of doing a lot of this for us. So let's look at the first one, which is MCV, mean corpuscular or mean cell volume. A corpuscle is just another name for a red cell. And this looks at the actual volume or size of an average cell, one cell. Are they bigger than normal, smaller than normal, or average size? Mean cell hemoglobin tells us the weight of hemoglobin that is in each cell, so how much is physically there. Mean cell hemoglobin concentration tells us the density of that hemoglobin within the cell, so how concentrated is the cell with hemoglobin. Oftentimes the color will help to clue you in on this because the darker it is, generally the more hemoglobin it contains. And then the red cell distribution width is a little bit different because it is looking at the whole population of cells in the sample and how much they vary in size. And what you'll get is you'll get a picture on the screen of a histogram, and if it has a really wide range, that indicates that you have a lot of cells that are many different sizes. Some are really small and some are really big. These are all red cells, mind you. And if that happens, then we know something's going on that's sort of strange with the production of the red cells. So again, in RDW, you're looking at the degree of variability of the red blood cell size. So in this case, we're not really looking at an individual cell, we're looking at the whole population. But we're going to talk more about these in class, and you'll be doing a little bit of reading on the math behind these different indices as well. So our goal was to explain basic hematology procedures, and the ones below, as you can see, we've talked about the complete blood count, hemoglobin measurements, hematocrits and packed cell volumes, and the red blood cell indices. If you have any questions, you know where to find me. Feel free to email me, stop by my office, or ask in class. Thanks!